to download a smell. We are not there yet. But the first step is to digitally analyze a smell. And that is what Charlotte Dulst is doing with her New York-based company, Mouse Sensor. I'm sorry if the next sentence sounds a bit like Walt Disney, but here it is. Mouse Sensor engineers super sniffer mice to build a nose on a chip. Super sniffer mice, nose on a chip, to digitize every smell on the planet Earth. Ladies and gentlemen, I smell a hint of success. Here is Charlotte Dulst.
Good afternoon, everybody. Even for New York standards, this is a pretty impressive stage, so bear with me. So this is Joy. Joy noticed a particular smell in her husband's sweat when he was 33 years old. He got diagnosed with Parkinson's disease at age 45. It was only when Joy and her husband Les started going to these Parkinson's support groups that she noticed that all patients in the room had that same characteristic smell that she'd been smelling for over a decade. Now, these stories of sniffing disease are rather anecdotal than driving medical research. What if we could use our sense of smell for diagnostic purposes? Smell is an entire field of data that nobody has gone after because we have no objective means to detect, to characterize, and to index odors. When Google was founded in 1998, it was in their mission statement to organize the world's information and make it universally accessible and useful. This kicked off the digital revolution. In 2018, it is our mission to open up an entire new world of data and to establish the first ever digital database of smell, the Shazam for odors. The mouse sensor platforms allows us to translate the biology of the real nose onto a silicon surface to create a true biological nose on a chip. This nose can be odor-specific, like Joy, but on a chip, or by putting the full human nose on a chip, we now have the tremendous opportunity to finally decode the last mystery of the senses, the sense of smell. All unique odor codes that we will capture with our chips are then indexed into one odoromics database. And it goes without saying that the more data we feed into this platform, the smarter our noses will get. So the big question is, why? Why would somebody want a digital code for smell? And this brings me to a very interesting definition of innovation that I read in a book called Disciplined Entrepreneurship a while ago. Here, the author claims innovation is a product of invention and commercialization. Because it's not just about the idea, it's about making the idea happen. So let's start with why. So we actually got out of our comfort zone of the scientific lab and started talking to real people to find out what is their problem, because nobody is looking for a solution to a problem they don't have. And surprisingly, we found that there's actually two very different industries that have a major interest in a digital code for smell. So let's start with the fragrance and flavor industry, which is much more than just fine perfumes. Every smell molecule created by the 5 major fragrance houses on this earth are incorporated into every aspect of our daily life. Yet fragrance discovery is highly inefficient. The fragrance houses spend about $1 billion a year on their fragrance R&D pipeline, which is entirely based on trial and error. They don't have an objective means to measure a smell of an odor. Instead, they use people. They use trained perfumers, l'aîné, who are trained for 7 or more years to become highly efficient in assessing the smell of a new molecule. These are the rock stars of the industry that are often paid more than the CEOs of their companies. So what they need is a digital code for smell to, for example, predict how a new-to-the-world molecule synthesized by their chemist will actually smell. Take, for example, the smell of a rose. Apparently, the best rose smell out there is coming from roses that are freshly picked on Bulgarian fields at 5:30 in the morning. If they were to have the digital code for that particular smell, they would be able to reformulate the molecule in a much more sustainable way. We also talk to people in the healthcare industry. Now, the sense of smell has been used for ages in the practice of medicine. The ancient Greeks used their noses to smell tuberculosis. Currently, there are dogs that are better at sniffing out cancer than any medical diagnostic that's currently on the market. And then we have Joy, who is able to detect Parkinson's before onset of the symptoms. So what if we could use our nose-on-a-chip technology to create an entire new field, an entire new science of odor-based disease diagnostics? So the next question in our innovative journey is how? How are we going to do this? And bear with me, because I'm going to try to make this as simple as possible, but not simpler. So when odors enter our noses, they bind with an array of sensors in our nose, and each odor has a very specific combination of sensors that they activate. Now, the problem is that this is what we call the combinatorial code, and for most odors, this combinatorial code is completely unknown. The thing that we do know is that there is a vast repertoire of genes in our genome that is responsible for making these odor sensors. The human genome has about 350 different genes coding for odor receptors. The mouse genome has about 1,200, and elephants have about 2,000 genes that code for odor sensors. Now, the big problem that our field of olfaction has been facing for the last 25 years is that these odor sensors, which are basically proteins, cannot be made in a dish, in vitro, in a lab. So that's why we were never able to make these big odor screening assays to actually decode olfaction. So at Mouse Sensor, we argue, why trying to make a better receptor in a dish artificially, or why try to mimic these receptors electronically, why we can just use the best ones out there that have been fine-tuned to millions and millions and millions of years of evolution. And that's exactly what our mouse sensor platform does. So the mouse sensor technology is a genetic way to turn the nose of a mouse into a bioreactor for very functional odor sensors, which are produced in their natural environment, the olfactory sensory neuron. We pride ourselves that this technology is rooted in basic science. The nice thing about using genetics to make this platform is that we can label the sensors that we clone into our platform with different fluorescent proteins, shown here in red. So we can actually follow their expression both in the nose as well as in the brain. The past 2 years, we worked on a technology to translate this validated in vivo model to an ex vivo model so we could actually use it for high-throughput odor screening. So we found a way to extract these very specific odor sensors created in the factory of a nose out of the nose while keeping their odor sensing machinery completely intact. And interestingly, we can also freeze these, what we call bioextracts, at -80 degrees without losing their activity, which is very important if you want to scale your model to a very high throughput system. So recently, we partnered with what they call the smartest square kilometer in Belgium, IMEC, to further miniaturize our assay from a 96 well plate to a nano chip. Herefore, we re-engineered our animals to have, instead of these red reporters you see on the left, a green reporter. And the beauty about this green reporter is that if the sensor gets activated by a specific odor, it emits fluorescence, which we can detect with a microscope. And this way, our system is now amenable to optical detection. So fast forward, how will this chip look like? So you can imagine this chip being a CMOS chip or an optical chip that you find in your cell phone. We then might make bioextracts that represent every single odorant receptor in the nose of a human, and we put them on the different spots in the chip. Then we add an odor through, for example, microfluidic systems, and the odor will activate a very specific subset of receptors on the chip, and only those that get activated will actually emit fluorescence, which will then lead us to the code for that particular odor. Now, that said, we are still very early stage. So we have currently, or we just finished raising our first seed round to make the prototype powered by IMEC. And we are very grateful for the support that we received from the IMEC Expand Fund and also an American investor, Alexandria Ventures. Now, we were only able to survive the Valley of Death, which is also called the dark swamp of despair, by having funding sources that are not coming from venture capital. Because that is the most important thing in the beginning. When you spin out a technology rooted in basic science from a university, you have to have money to further establish the R&D, but you're still at a much too high technological risk to actually be attractive for venture capital. So one of our very early supporters was actually the Michael J. Fox Foundation for Parkinson's research, because they saw important potential for the mouse sensor technology in potential early detection of Parkinson's disease. So we set out with the Michael J. Fox Foundation on this adventure to actually identify the combinatorial code, the specific subset of odor sensors that get activated when exposed to Parkinson's disease. So herefore, we work together with Joy Milne, shown here with her husband Les. And so Joy was asked to validate and to smell T-shirts that were worn by Parkinson's patients for 24 hours to collect the sweat on the upper back. These T-shirts were then exposed to rodents, shown here, our little rats, and we used a specific genetic technique to read in the rodent's genome what specific set of odorant receptors actually got activated when exposed to the T-shirts that were drenched in Parkinson's sweat. So here we identified an initial candidate combinatorial code for Parkinson's disease, for which we have now made our first mouse sensor animals. So in this case, we chose to label these Parkinson's code with, or these Parkinson's receptors with teal and with yellow, as you can see here in the brain of one of our mouse sensor animals. This here is a section through the nose of these animals, and all the little dots in teal and in yellow represent receptors that are potentially activated by Parkinson's disease. And this model is currently further studied in our lab in New York. So with this, I really want to thank my team who make the magic happen every day, both here in Belgium at IMEC, as well as in our scientific labs in New York City. Mouse Sensor is headquartered in New York. We are tenants of the Alexandria Launch Labs incubator in the heart of Manhattan. So please come say hi when you're in town. Thank you.